All right, so I've got news. So we'll go ahead and get started. Firstly, I want to thank you all for coming out today. This is our last Bear Ranch Foundation seminar of this semester. We've got a good lineup for next semester, but we'll send that around to all of you once we've got that finalized. Also, for the graduate students, lunch will be in 138 afterwards, so you can meet with Dr. Decker, have lunch. It's free for you. So with that, it's my pleasure today to welcome Dr. Jared Decker. He comes to us from the University of Missouri, where he's the WordAct Chair in Animal Genetics and Genomics. Prior to taking that position on earlier this year, he was serving as an extension specialist there in beef cattle production, and his research program focuses on utilizing quantitative genetics and population genomics and trying to merge the two to give us a better insight on how selection is impacting our cattle populations. He is pretty much a straight Missouri graduate, where he did his PhD as well as did a short postdoc there before being hired on as faculty. Prior to that, he is originally from New Mexico, where he did his bachelor's degree at New Mexico State there in animal science. And today he's going to talk about a little bit of his research, and we're glad to have you here, Dr. Decker.
Thank you. Thanks for that great introduction. So, I'm really happy to be here. Really appreciate Chris being such a great host. I think your department is very similar to the department that I did my undergrad in at New Mexico State, where we had both range and animal sciences put together in a single department. And so when we're talking about the various topics that I could talk about today, this is kind of what we settled on because we wanted to touch on things that where genetics interacted with nutrition, where genetics interacted with environment and those type of things. So as Chris mentioned, my lab blends both evolutionary biology in the form of population genomics with quantitative genomics. So basically, I joke with people that what we do is basically 23andMe in cattle. And if the conversation continues, I can explain to them that what we do in cattle is actually works better than anything that 23andMe sells. Also, to start out, I want to thank my many collaborators. Bob Schnabel is an associate professor at the University of Missouri. He and I have joint lab meetings together, do a ton of work together. In today's presentation, I'll present quite a bit of things that Harley Durbin, Troy Rowan, and Sarah Nielsen have worked on. Harley and Troy have graduated now. And then this is my current group there at Missouri. Also want to thank Semitole Association, Red Angus Association, Angus Genetics, and then a lot of the data that I'll talk about today is data that we have from the American Gelbvieh Association. All right, so anyone who has ever tried to communicate the science of EPD, farmers and ranchers, knows that there's a lot of mistrust of that technology in the beef industry relative to other more vertically integrated protein like poultry or swine, dairy industry. We have much less uptake of this old technology of EPD compared to other protein sources. So why is that? I think one of those reasons is that there's simply misunderstanding of how and why the technology works and what are the expected results of using the technology. But I think one of the other things that frequently happens in the beef industry is the animals simply underperform expectations. And this could be for lots of different reasons, but one of the potential reasons that I think could be there is we have genetic interactions with environment and management. So if we don't sophisticatedly account for and model these interactions and provide tools, more importantly, provide tools to allow farmers and ranchers to identify the genetics that are going to work best in their management, in their environment, then we can have these underwhelming results. A lot of times when we think about genetics and environment, and if we look at the literature, there will be tons of focus on heat, right? So the Angus animals in the swamp there, they're likely experiencing a high temperature and a humidity, right? It's hot there, it's humid there. The animals there on the range, it's hot there, but not nearly as humid. And so a lot of times when we think about environmental stress, people kind of get in their mind heat stress. But this cow here in the snowstorm is not experiencing any sort of heat stress, right? But we can look at her and easily see that she's under stress, right? So when I think about genetics and the environment, I'm really thinking about the entire suite of environmental stressors that those animals may be dealing with. And so it's really a lot more complicated and a lot more nuanced than simply. There's lots of things, parasites, cold weather, altitude, et cetera, et cetera. The other thing that if we just think, so I'm from Missouri in terms of where my professional life, and in Missouri we have this great forage fescue, and the predominant type is Kentucky 31 fescue, and it's super hardy to drought and raise it real hard and it comes back strong, but it has a fungus that lives inside the fescue. That endophyte produces a toxin that's one covalent bond different from LSD. So those ergovalent toxins have all kinds of stressors on cattle in Missouri. It causes vasoconstriction, which increases their heat stress because they're not able to dissipate heat as easily. And then it causes reduced feed intake, pregnancy outcomes, et cetera, et cetera. So all of these different environmental stresses cost the beef industry billions of dollars each year because animals simply underperform the ideal production environment. And so back in 2016, we received a very large grant from the USDA, and the purpose of that grant was to help farmers and ranchers develop breeds and lines that were more adapted to stressors in the environment, both abiotic and biotic stressors in the environment. So that's the title of this grant. And basically this was kind of the impetus of the work that I'll share with you today. Now, one of my favorite papers that's come out of my group is this paper that Troy Rowan has led. And basically what we do in this paper is we look at three different things, okay? We look at DNA variants that are changing in frequency over time due to the selection decisions of farmers and ranchers. Okay? So these are DNA variants that are changing in allele frequency due to that directional selection. Okay. The second type of DNA variant that we identified were, and when I say SNPs, I'm talking about a DNA marker. Okay. So we're looking for DNA markers that are associated with the environment. If a DNA variant is significantly associated with the environment, that association is likely caused by selection for local adaptation to that specific environment. And so in that paper, we were able to identify locations on the chromosome that were likely involved in selection for resilience in that environment. The third piece that we did in this paper was, instead of looking for DNA variants that were changing across the US, looked for DNA variants that were changing in frequency over time in specific regions of the US. And then we did a meta-analysis of these region-specific analyses and looked for SNPs that were changing in frequency in one region, but not the other regions of the US. And when we do this analysis, we can look at the allele frequency trajectories over time of these SNPs. And the common theme that we see from this third analysis in this paper is that back in the 80s, we had different allele frequencies in these different ecoregions of the US. So this is how we split the US into different ecoregions. And what we see is the allele frequencies converging towards the breed average. What we conclude from this analysis, and we never see the pattern going in the opposite direction. We never have allele frequencies at the same spot and then going to different frequencies in the different regions. Conclusion that we make from this analysis is that we're likely losing local adaptation to specific environments in beef cattle. And that makes sense if we think about the increased use of artificial insemination, moving cattle across the United States, the increased use of expected progeny differences that only focus on additive genetics. And so one of the things that when we talked to the press about this paper is we pointed out that farmers and ranchers simply don't have tools to select animals to match the environment, right? We do a really, really good job of providing farmers and ranchers to identify animals with better additive genetics. Identifying those animals with adaptations to environments, we haven't created those tools yet. So one of the ways that we can do that is by creating genetic predictions for new traits, novel traits, okay? So one of the traits my group has worked on is hair shedding. Hair shedding is when we make that transition from winter to summer, the cow shedding off her dead winter hair and hopefully having a shorter summer coat, okay? And it's a 1 to 5 scoring system. 5 would be that animal is still completely covered in that dead winter hair. She's still got dead winter hair on her neck and on her head. We tend to see these cows shed from the top to the bottom and from the front to the back. So a score of 4, that cow starts to shed off her winter hair on her neck. And what would distinguish a 1 from a 2? So a 1 would be a cow that has no dead winter hair left. And a cow with a score of 2 often has dead hair on her lower belly or her flank, and often on her lower quarter, okay? So why are we interested in hair shedding? I'm going to skip, this is just how we collected and generated the data. So we have data from basically 37,000 data points from about 13,000 cattle. We genotyped these animals and we shared the genotype information back with the breed associations, and the breed associations then provided the farmers and ranchers with 3 genomic enhanced DNA. So what we did is we said, give us your phenotypes and we'll give you 3 genomic enhanced partnership with the breed association. So Angus now has a research EPD on hair shedding. It's a combination of our data and data that they had funded that was collected by Mississippi State and North Carolina State. So that's available. You can get on the Angus website for, I think, the top, for bulls that have an EPD accuracy larger than 0.4, you see their hair shedding EPD. So why hair shedding? So none of us would want to walk around all summer with our Carhartt coats on, right? We would, especially if I'm in Missouri, I sweat a bunch in the first place. If you then make me wear a Carhartt coat, it's going to get pretty ugly pretty quick. The same thing happens with our cattle, okay? And so what we show here is data from 2016 through 2018, Semitol cows. On the x-axis, we've plotted the cow's hair shedding score. On the y-axis, we've plotted the calf's adjusted weaning weight. And we see that those earlier shedding cows tend to wean a heavier calf, simply because they're doing a better job. They're not expending resources on trying to deal with the heat stress, so they have more resources to put towards the calf. We also think that hair shedding likely has implications on fertility as well, and we're going to be looking into those in the future. Another example of a novel trait for local adaptation is patch, so pulmonary arterial pressure. They collect these patch scores, turn it into the breed association, and they collect and they create an EPD. This allows Angus producers to identify those sires and cows that are less likely to produce calves that have cardiovascular failure at high altitudes. Okay. However, we have lots of data available, especially a lot of growth data. So one of the other approaches that my group is doing is, can we analyze this existing data in different ways to better understand these genotype by environment interactions? So this is a Gelby data set, like I mentioned. It's about 12,000 genotyped animals, and we have about 8,000 to 10,000 growth records depending on the trait. Now, one of the things that we have to keep in mind is when we do genetic analyses, we adjust for what we call a contemporary group. And a contemporary group is simply a group of animals that were born in the same year, the same season, the same farm, under the same management. Okay? So in this kind of observational data, not all of the animals within a contemporary group are necessarily genotyped. Basically, the farmer's most common, what happens, the farmer's favorite animal from the contemporary group is the one that gets genotyped. Okay, so in this industry data, we have lots of small contemporary groups that makes it hard to adequately control for the effect of these management, these herd year season differences. So in this talk, in this research, we've adjusted for contemporary groups two ways. One, we fit contemporary group as a fixed effect in our model. If an animal is from a small contemporary group, simply fit the breed average. The other way that we can do it is with a random effect. Random effects are a lot better at handling this unbalanced data. And we pre-adjusted the growth traits for these temporary group effects. So our initial approach at trying to include environment, G by E effects into genetic predictions is using approaches from the 1950s. What they did there is the phenotypes, so the birth weight records from one environment would be one trait in our bivariate model, and the birth weight records from a different environment, the second trait in our bivariate model. What this allows us to do is estimate the genetic correlation between the birth weight performance in the context. And it also allows us to get a genetic prediction for both traits. So an animal has a birth weight observation, say in the high plains, but that animal also was not also born in the southeast. But because of the connectedness from the genetic similarity from the genomics and the pedigree information, that animal actually gets a breeding value in both regions. However, we've kind of abandoned this approach because if you've got 9 different ecoregions across the US, you have lots of different combinations of environments. And so that you end up with lots and lots of different models that you're trying to fit. The other thing that we ran into is models tend to have very poor convergence. It was hard to get the model fit properly. But here's some results from some of those that we ran. This is birth weight. And ecoregion 3 here is the high plains. Ecoregion 9 is kind of the upper Midwest and the Northeast. And you can't quite see it, but over here, the prediction accuracy, if an animal was in the high plains and we predicted their performance in the high plains, that prediction accuracy was about 0.6, right? Or if we compared it to their pre-adjusted phenotype, that prediction accuracy was about 0.70, okay? If an animal was from ecoregion 9, so the upper Midwest and Northeast, and we predicted their genetic merit in the high plains, that accuracy was about 0.6. However, we have very few animals, relatively fewer animals. We had about 1,000 animals in the high plains, about 1,000 animals in the upper Midwest. When we tried to predict animals from the high plains for their performance in the upper Midwest, that prediction accuracy was quite low. So this just kind of highlights the difficulty in getting these bivariates work well. And so that's driven by the fact that you're taking your large data set and splitting it up into much smaller data sets. And genetic prediction is driven by the size of your data, right? Anytime we put more information and more data into a genetic prediction, it improves. So cutting the size is problematic, and predictions tend to be less accurate. So we can switch to a different approach, and we fit what's called a G by E genomic relationship matrix. And what we do here is we take our genomic relationship matrix. So let's say in the Gelby data, we've got 10,000 animals, right? 10,000 birth weight records. This matrix is 10,000 animals by 10,000 animals, right? 10,000 rows by 10,000 columns. What we do to create a G by E GRM, if 2 animals are in the same region, we simply copy over their genomic relationship value, okay? But if 2 animals, one animal is in region A and another animal is region B, we then set their genetic relationship to 0. So what we do with this genomic relationship matrix to look at the interactions between genetics and the environment is we look at additive relationships within an ecoregion, and then we compare and contrast the different regions of the US, compare and contrast the different environments. So what we've done here is we've estimated the variance components in a software called GCTA, and then done the breeding value prediction in a suite called BluffS90. So when we do this with birth weight, what we've handled contemporary group 4 different ways here. Okay, we've either not adjusted for contemporary group. That's not quite appropriate, but it's shown here just for illustrative purposes. We fit contemporary group as a fixed effect, contemporary group as a random effect, and then we've included a maternal effect, not a genetic maternal effect, but simply effect, a random effect of who the did. Okay. And what you'll see is as we more sophisticatedly adjust for the contemporary group, our heritability estimates increase. Okay. So as we better account for management differences, more of the variation that remains is additive genetic variation. However, we more sophisticatedly adjust for management, genotype by environment variance goes down and becomes a smaller and smaller proportion of the total phenotypic variance in the population. So one of the take homes here from this is that we've got this tangled mess, right, between management effects and environment. Okay? And that's going to be one of the reoccurring themes in the results that we share with you all. Weaning weight, we see much of the same things going on. Again, as we more appropriately adjust for those contemporary group effects, the variance associated with genotype by environment interactions goes down. But even at its lowest, that genotype by environment interaction is still accounting for about 6% of the variation in the trait. So it's not like it's minuscule and non-existent. It's there and is something that we can use to allow farmers and ranchers to make more informed decisions. And then here's the results for yearling weight, heritabilities, and then the proportion of the variance due to genotype by environment. So like I mentioned, we've got kind of this confounding that happens between management and environment, right? And we can think about this in the extremes in other livestock species, right? So if we think about broilers or swine production, those animals are housed in a confined building where we're controlling the temperature of the building, right? So we're doing a lot of management to try to manage around the environment. We have those same sort of dynamics in the beef industry as well. If, say, an animal is struggling with the environment, we might increase their supplementation. The other thing that I want to point out is these maternal effects also appear to be important. So if we think about who's actually dealing most frequently with the environmental stress, it's the mothers, right? It's the dams, it's the cows. And so really one of the, okay, so this is a different paper that we've published in Genotype panel. And in this, we were doing lots of different analyses, looking for individual DNA variants that were associated with genotype by environment interactions. We see about 384 significant genotype by environment interactions with birth weight. When we go in and account for maternal effects, 94 of those interactions effects go away. So a lot of what we're seeing is the cow is dealing with some sort of environmental stressor, and that is then getting expressed in the birth weight of her calf. Really, so in future directions, as we move forward with this line of research, really where we'll go is fitting not just random maternal effects, but maternal genetic effects, okay? And then as we fit those maternal genetic effects, we'll be fitting maternal genetic by environment effects as well. And we think that this is where a lot of those genotype by environment interactions are having an impact is on the dam of the calf and how her either being resilient or struggling with the environment is affecting the performance of the calf. So those will be more sophisticated models that will be fitting into research. Okay, now back to that Gelby data and looking again at birth weight. These are the accuracies of prediction for a national cattle evaluation model. Okay, so we take all 10,000 Gelby and we analyze them in one model, and we really don't do much to try to account. We don't do anything in these models to look at genotype by environment interactions. So whether we compare breeding values to breeding values with what we call the LR method, or we compare an adjusted phenotype to a breeding value, those prediction accuracies are in that 0.7 to 0.74 range, okay? Now in these models here, here's one model where we've pre-adjusted the phenotypes for fixed contemporary group effects, or where we pre-adjust the phenotypes for random contemporary group effects. What we can do is we can compare this to this, and we see here for birth weight, for most instances, we don't see much of an improvement, and in one case we see a small decrease in the prediction accuracy when we account for these GIE effects. The other thing that I'll point out here is that these G by E effects are quite difficult to predict accurately. It's much easier to predict an accurate additive genetics breeding value than it is to predict these G by E random effects. Switching now to weaning weight, again, for the national model, fairly good prediction accuracies, and then we can break it apart. And one of the things that I'll point out here is as we compare the phenotypes to the breeding values in a national model versus the phenotypes and the breeding value in a model that also includes G by E, is we see a fairly substantial increase in the prediction accuracy of those additive genetics. Okay, so this might be a fruitful area where we can squeeze more information out of our available data and produce more accurate predictions for farmers and ranchers by better accounting for those genotype by environment interactions. But those G by E effects are still fairly difficult to predict accurately. And yearling weight, same trends again, pretty good prediction accuracies for a national model, and we see small bumps in improvement for the models in which we include G by E effects. So this is just a graphical representation of those previous two plots. Got the partial breeding value versus the whole breeding value. This is from that LR method. We see a nice slope, so we don't have any bias in terms of over or under dispersion of our breeding values, and see nice genetic correlations. But as I kept pointing out to you, it's fairly difficult to get good predictions of those G by E effects. And for some reason in this data set, we predict a whole bunch of animals to have a G by E effect of 0 when their true G by E effect is quite different from 0. So that's some sort of phenomenon that's happening in these data. I don't understand why that is, but this group of animals here that we're simply failing to predict accurately is probably a main driver for why those G by E effects are less accurate. So some of the next steps that we're going to do is I'm still convinced that we need to create tools for farmers and ranchers so they can actively select cattle that are matched to their environment. In terms of this work, we'll work on creating for the additive genetic effects, instead of using all 830,000 DNA markers, we'll enrich it just for the 20,000 DNA markers that are changing in frequency over time due to selection. We can create that G by E GRM, and there also, instead of using all 130,000 DNA markers, we can select those 20,000 DNA markers that have evidence for local adaptation selection or other G by E effects. And what I'm really excited about is in this Gelby data, as you've seen, we have about 10,000 animals. In the Simmental and Red Angus data sets, we'll have a little less than 100,000 animals and a little less than 50,000 animals. And so this will allow us to to filter out those animals that are from the really small contemporary groups and really have more power to look at this data and these phenomenons in a more powerful data set. So really excited to see how that works out. So visiting this message again, we have this confounding between environment and management. And one of the things that we've seen both in some of our Red Angus data sets and in our Angus data sets is when we look at weaning weight contemporary groups, when we look at birth weight contemporary groups, we have this nice normal bell curve. But when we look at the contemporary group effects for yearling weight, we see this bimodal distribution of the contemporary group effects. Now, one explanation that I have for this is this simply reflects low input type management, perhaps yearling development on forages versus higher input management where we're developing those animals on concentrate. And so basically what we have is a mixture of two different distributions of two different management scenarios. And so one of the things that we've realized through this genotype by environment work is perhaps what might be more important is actually trying to account for genotype by management interactions. And so we can still include those G by E effects, but also include a G by N effect as well. So we have a new grant funded to continue this work. In evolutionary biology, there's this rule called Bergman's rule. And so as we look at the equator and we move north and south from the equator, a common observation that we see is that animals get larger the further from the equator that we get. There's two competing hypotheses for this observation. The first hypothesis is as we get to these colder and colder climates, this larger body size has a smaller surface area to volume ratio. Small ball or a big ball. The big ball has the smaller surface area to volume ratio. Should allow that object to retain heat more easily. Okay, so that's one hypothesis for this Bergman's rule observation. The other hypothesis is, as we move further from the equator, we have this big pulses in primary productivity of the plants that these animals are eating. And so basically what it's saying is we need bigger animals to utilize those storage resources as they come along at these higher latitudes, further away from the equator. So basically what we're talking about here in terms of the way I framed it in the talk today is we're talking about genotype by environment effects, right? Being able to retain heat versus genotype by management effects, the ability to utilize nutrition supplied to the animal. So we're going to measure surface area and volume ratio of thousands of animals, and we're going to compare those to our genotype by environment effects and our genotype by management effects to try to answer this question of what's really driving Bergman's rule. Okay. And what we'll also in this project create tools for farmers and ranchers to select better cattle. Okay. So as we think about sustainability, think about producing cattle more profitably, producing cattle with a smaller input on the environment, producing cattle in a way that society feels good about the welfare of those animals. These genotype by environment interactions and genotype by management interactions come into play. And I think as we try to increase in efficiency, we'll see many producers turn towards having lower inputs to increase the profitability and efficiency of their operations. And so I guess the question that I have, and I'll leave you with today, is as we adjust our management practices to be more sustainable, do we have the cattle genetics that will work under those contexts? So that's really the big picture problem that my group is trying to work towards, is creating these genetic tools to allow farmers and ranchers to select the genetics that match their environment, select the genetics that match their management. Point you to several publications from our lab over the last year or so. And again, just thank our many collaborators. With that, I'd be happy to take any questions you have. By easy by animal. Breed. Yeah, so I think, you know, in the plant world, kind of the big complicated models that they're thinking about is genotype by environment, by management, by social effect. Kind of ignored that social effect type. So could be worried about type by environment, by management action. But I think before we get to that level of complexity, probably need to go through the steps of having the more simple models where we first look at genotype by management. So it'll kind of be a stair-step approach, you know, little by little, trying to add to and the models. The other thing that happens is increase the complexity of the models. it gets harder and harder for the model to actually converge, for the model to get to the solution. So I think that's another reason why we may err towards, good question. Right. Great question. So that was part of the reason we started with multivariate models. Got a reading value, one piece of information that had the genotype, that had the additive genetics, variety effects put together in one number for the farmer and rancher. Like I talked about, those models don't converge well. They've not been a good tool for us to use. So I think perhaps the next step is simply to fit the G by E effects in our model. And what that would allow us to do is get EPs that are simply more accurate. And so the tool that we're providing to farmers and ranchers is more accurate. And perhaps we're not providing them with the G by E effects. We're simply giving them a tool that's better because we've more sophisticatedly analyzed those G by E effects. The problem with the way these models are set up is animal gets a deviation basically from its environment. Okay, so for animals that had phenotypic observations from the high plains, we could say this animal outperforms the average of the high plains, or this animal underperforms the average of the high plains. But that animal's phenotypic observation in the high plains doesn't tell us anything. The way we set up the model, that deviation from the high plains doesn't tell us anything about how their progeny would perform in the fescue belt in the southeast. So how we actually get to the point where we've got better actionable data to match those cows to the environment, I've not fully wrapped my head around that yet. The other approach that we commonly see will have random regressions, and usually it's back to that heat stress thing where they're fitting a index of heat and humidity, but then it's back to kind of the first point I made in the talk of we get kind of myopically focused on heat and humidity and miss all of these other things going on. So perhaps one possible solution there is instead of fitting these G by E effects the way that I have, is we take all of the environmental data, do a principal component analysis of all of the animal's environmental data, and says, here's the largest principal component, and we fit that principal component that's taking into account the elevation, the precipitation, the temperature, so on and so forth, as the variable in our random regression. And we could look at it that way. So then we could say, if you're at one end of the environmental extremes, here's your EPD. You're at average of this random regression, here's your EPDs. If you're at the other extreme, here's your EPD. Still kind of an open question. I've not personally figured out what is the best way to get to that. So I think that's also where perhaps these novel traits come in, is it's something easy to record, easy to analyze, to give people, an easy select for environmental wisdom. Um, so, you're not gonna like this answer. I've sent a document to my college, to my university's IP office, and I'm not supposed to talk. So we've got ways that we're going to do it. They're evaluating the commercial application of those methods. Hopefully in February, I'll be able to talk about it publicly. Yes.
Make sure
Yeah, if you if you if you get on USDA's website, you can find the records for that Bergman's rule and cattle grant. Just to keep my IT people happy, I'll be paid, so apologize for that. Yeah, so one of the things that we'd like to look at, we talked about this, both with Semitol and with Earthbird, is comparing, whether a cow got pregnant or not to her hair shedding score. Fitting basically a bivariate model with those pregnancy outcomes with hair shedding. So yeah, so the growth data that I talked about, that's obviously one impact is cows suffering, so the calf suffers. But probably the impact that's gonna have the bigger economic consequence is if the cow's struggling with heat stress and so she fails.
But like a cow, ecoregion. Lot of variation. That sort of thing. Traumas and
Right, right. No, so there's been people that have done that. And so what they do in that approach is they go through and they fit one model. They get the contemporary group solutions. They then use the contemporary group solutions as the variable to use in their random regression. And so what you end up with there is both the year to year, season to season, management effects, but then you also end up with the differences in management between those contemporaries. So a lot of times when people fit those temporary group solutions as the regressor in a random regression, they're conflating that environment and management. So it's a good approach. One of the other things that I worry about with that approach is we're analyzing the phenotype twice. So I would worry a little bit about overfitting or something like that. So yeah. So one of the things that I'm excited about in this is I think there's lots of open questions and there's no one clear solution about how we make it work. But then the dissatisfying thing is we don't have a clear solution. One of the things that we're doing as we're starting to create these, what we call a management relationship matrix is, instead of keeping the contemporary group definitions very broad, so year, fall versus spring, etc., is we're trying to get a little more nuanced. Was the calf born in January? Was the calf born in February? Was the calf born in April? Because in my mind, if you decide to calve in January versus if you decide to calve in April, those are very different management decisions. And so trying to look at that kind of when in the year they're calving to look at that management. But then again, it's still kind of, it's all tangled together between, that calf born in January is dealing with a much different environment than that calf. Yes, so, I've got similar questions before. Could we simply, another way I've heard that question phrased is, can we simply change our weighting in our economic selection index based on the environment? And the problem with that is these are not linear differences. These are non-linear differences. And so it's not going to be as simple as a linear adjustment factor. So I think, there's some animals that are pretty average across all the different environments. There's some animals that perform really well in one environment and really poor in a different environment. So because we have these very non-linear effects, I don't think it's as simple as identifying the right adjustment.
Along that line, as I was thinking. Very much. Area.
Right.
That's but they just published high elevation. That's how they.
Yeah. And a different way to perhaps frame that approach is they're using those lower elevation CAP scores as an indicator for high elevation. It's a little bit like how, one way you could frame that is like how we use birth weight to better predict calving. But yeah, yeah, it is interesting that it does kind of have that key by income.
Down the hall, 138.